Hello students, myself Abhikumar Sharma, Assistant Professor in Agriculture Department at GGDSD College Haryana. Now today's topic of lecture is nucleic acid. I repeat, today's topic of lecture is nucleic acid. Nucleic acids are basically, very well known for DNA and RNA. Nucleic acid includes DNA and RNA. DNA and RNA basically these are biopolymers. Biopolymers means they are living polymers present in living organisms. So polymers, poly means number of monomers. What type of monomers are present in these molecules? The monomers are nucleotides. So later we will discuss the composition of nucleotides. These are large biomolecules. This means they have macromolecules having large in shape and structure. Nucleic acids are essential for life. As we all know, these are the hereditary material that get inherited from parents to the offspring. So nucleic acids are very essential for the continuity of life. These include DNA and RNA. Full form of DNA is deoxyribonucleic acid and second RNA ribonucleic acid. The main difference between DNA and RNA is the presence of pentose sugars. In the case of DNA, the pentose sugar is deoxy, but in the case of RNA, the pentose sugar is ribose sugar. Made from monomers, as I already told you, monomer of DNA and RNA are nucleotides, known as nucleotides. DNA first found in the nucleus of the cell. As we all know, cell consists of cell membrane, then in the center there is a presence of rounded nucleus. Nucleus contains nucleic acid, that is DNA or RNA. So first found in the nucleus of the cells. Second, later on discovered outside the nucleus. DNA also present in the cytoplasm. As we all know, cytoplasm contains number of organelles. Out of that organelles, the two organelles are responsible. Those contain nucleic acid. Out of them, the two organelles are mitochondria and chloroplast, over which DNA is present. So this type of DNA or genes present in the cytoplasm are called polygenes or cytoplasmic genes. Next, history of nucleic acids. In 1869, Friedrich Mischer isolated intact nuclei from cell and analyzing chemical content. In 1869, Friedrich Mischer isolated nuclei. He just isolate nuclei from a living organism or from a living cell, and he analyzed what type of chemical contents are present within the nucleus. So what he got the extracted substance were rich in phosphorus and nitrogen, which was known as nucleic acid. After this discovery, he gave a name, that is nucleic acid. In 1944, Avery, MacLeod and McCarthy, were the three scientists who also did work over the nucleic acid or in the isolation of nucleic acid. They expressed it as DNA and found as a genetic material. These three scientists, what they conclude, DNA act as a genetic material, by using bacterial DNA to transform the genetic material of other bacteria. Or they also give the idea about the rDNA technology, where the bacterial or plasmid DNA used for the transforming of existing DNA into a recombinant DNA. Next slide is composition of nucleic acid. Next slide is composition of nucleic acid. As I already told you, nucleic acids are DNA and RNA. These are the polymers that is made up of monomers. So the monomer of DNA and RNA is nucleotide. Nucleotide mainly composed of three components. These are nitrogenous bases contain purines or pyrimidines. Second, pentose sugar, that is 5 ring structure. Third, phosphate group. Third is phosphate group. All three components combined with different type of bonds to make a single monomer known as nucleotide, and number of nucleotide joined by phosphodiester bond to make a long chain of polymers of a DNA or RNA. Clear? Now next, composition of nucleic acids. Again, nucleic acid polymers, polynucleotides, even polymers, you can say DNA, also known as polynucleotide, forms from the nucleotide monomers when the phosphate of the one nucleotide bonds to the sugar of the next nucleotide. This bond is known as phosphodiester bond. This bond formed between the phosphate of one nucleotide with the sugar of the next nucleotide, resulting a repeating sugar phosphate backbone with producing nitrogenous bases. In this continuous manner, from 5 prime to 3 prime, a long chain of deoxyribonucleotides is formed, and same in antiparallel form, second chain is from 3 prime to 5 prime. Structure of nucleotide, you can see here the nucleotides is of two type, purines and pyrimidines. Purines are adenine and guanine, and pyrimidines contain cytosine, thymine, but and third is the uracil. The main difference between DNA and RNA, the thymine is replaced by uracil in the case of RNA. Rest nucleotide remains same. The difference is only between RNA and DNA is the nucleotide thymine is replaced by uracil in the case of RNA. You can see here a yellow rectangular shape structure showing phosphate group. Then in pink there is a pentose sugar. Then in the light brownish block showing deoxyribo rings of a sugar. Next, type of nucleic acids. As I already discussed, type of nucleic acids are of two types, DNA and RNA. DNA means deoxyribonucleic acid, double stranded. This structure is double stranded. This means there is a presence of two strand. One is from 5 prime to 3 prime and another is 3 prime to 5 prime. Helicals means it is just in the twisted form. These two strands are twist right-handedly. The polymers of deoxyribonucleotide found in the nucleus of the cells, as we all know it very well, DNA is always present in the nucleus of the cell. What is the function of DNA? Storage of the genetic material. it acts as a genetic material, or it stores the whole genetic information of organism within it. Second is ribonucleic acid, that is RNA. RNA is always single stranded. There is presence of only single strand, that is 5 prime to 3 prime. No any antiparallel strand, no any complementary strand. It is always in a singular form. Polymers of the ribonucleotide found in the nucleus and the cytoplasm of the cells. Mostly RNA found both in nucleoplasm and also in cytoplasm as well. Function: to transfer the genetic information from nucleus to the cytoplasm for the protein synthesis. The main function of the RNA is to transfer RNA or genetic information from nucleus to the cytoplasm for the further translation process, which leads to the formation or leads to the synthesis of proteins within the cytoplasm. Further, on the basis of this, RNA divided into three types: mRNA, that is messenger mRNA, rRNA, that is ribosomal RNA, and tRNA, that is transfer RNA. Messenger RNA is the complementary copy of the selected region of the DNA. It carries the genetic message from the nucleus to the cytoplasm and act as a template for the protein synthesis. It just encode the information that is complementary to the DNA for the protein synthesis. It helps in the process of transcription. It just copy information from the DNA and change it into the RNA. Next, ribosomal RNA. Ribosomal RNA basically used for the formation of ribosome. Ribosome contain one large subunit and one small subunit. These ribosomes are the main site where protein synthesis takes place. The function of RNA is RNA along with the protein forms of the ribosomes, which is the site of protein synthesis. Some of them have catalytic and coenzyme function as well. Some RNA also act as a catalytic, that is, they have a capacity to break down a large molecule into the smaller ones, and as a coenzymes function as well. So it RNA also act as a coenzyme. Coenzyme is a part of the holoenzyme. Third is the tRNA. tRNA is known as transfer RNA. Its main function is transfer the amino acid from the cytoplasm to the site of protein synthesis. As we all know, proteins are made up of amino acids, so tRNA helps in the proper binding of amino acids with the help of peptide bonds in a proper chain for the proper synthesis of protein within the cytoplasm. So next is biological role. In biological role, nucleic acid having a great importance in pharmaceuticals also. These nucleic acids are used in cancer, cardiovascular, and autoimmune diseases. The pharmaceuticals perspectives of nucleic acid based therapy presents a comprehensive account of gene therapy. These nucleic acids are very helpful in gene therapies. And next is internationally acclaimed scientists discuss the potential use of lipids, peptides, and polymers for the vivo delivery of nucleic acids. Vivo means, that is, vitro means within the lab, vivo means out of the lab. If anybody wants to take any nucleic acid, so they can get it even out of the lab also. So these are internationally acclaimed scientists discuss the potential use of lipids, peptides, and polymers for the vivo delivery of nucleic acids. control gene regulations, transcription, translation, and replication. As we all know, as per central term of science, DNA formed from DNA is known as replication. Then formation of RNA from DNA is transcription, and formation of or synthesis of proteins from RNA is known as translation. All these processes occur within the cell of our organism with the help of these nucleic acids. So next are the function of nucleic acids. Here we will discuss the function of DNA, that is deoxyribonucleic acid. The first function of DNA is permanent storage place for genetic information. The whole genetic information always stored in the DNA. All gene combinations, all type of genes, all type of genetic information is encoded or stored in the DNA. Second, controls the synthesis of RNA. As we know, RNA is formed with the help of transcription from DNA, so it also controls the synthesis of RNA. Third, the sequence of nitrogenous bases in DNA determines the protein development in the new cell. The sequence in which the genes are arranged in the DNA, they ultimately produce proteins or sequence of amino same as it is of the sequence present in the DNA. Next, the function of the double helix formation of DNA is to ensure no disorder occur. As we all know, there is a presence of hydrogen bonds between the nitrogenous bases, that is triple bond between adenine and guanine, and double bond between adenine and cytosine, triple bond adenine, guanine and cytosine, triple bond adenine and thymine, double bond. These bonds are very strong bonds, so they ensure no disorders to be occur. Next, this is because the second identical strand of DNA that runs antiparallel to the first is the backup in the case of lost or destroyed genetic information. So the second identical strand of DNA always help to to make a new complementary strands. As per the rule, adenine is always bind with thymine, and guanine also always bind with cytosine. Example, Downey syndrome or sickle cell anemia diseases. Then next function, we will talk about the functions of RNA. Ribonucleic acids are synthesized by DNA for the transportation of genetic information to the proteins building apparatus in the cell. So RNAs are the main biomolecules that are responsible for the protein synthesis within the cell. Synthesis of new protein by using genetic information it has transported. So new proteins using genetic information of RNA as a source for their synthesis. mRNA, as already discussed, messenger ribonucleic acid, is used for transferring genetic information through the plasma membrane, and RNA also directs the nucleic acid, especially DNA, carried out a vital role in the human body. In particular, nucleic acids play an essential role in mitosis, meiosis. As we all know, the cell division, having two type of cell division, that is mitosis, where chromosomal number remains same 2 and get divided into two daughter cells having same chromosome number, that is 2 and 2 and. But meiosis, that is a reproductive cell division, you can say, or gametic, where gametes are produced through this process, where the chromosome number get half from diploid to the haploid number. And second, providing energy and cellular respiration. So proper nucleic acid providing energy in the form of ATP, then that is adenosine triphosphate, and cellular respiration, proper cellular respiration, that is intake of oxygen and removal of carbon dioxide with the liberation of energy in the form of ATP. So these are the some references that I had used. So next, thank you very much. I hope you all got my points, I discussed, or whatever I taught you in this lecture. Hope all the best. Thank you very much.